Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Enzyme Manufacturing and Optimization via an E. coli One-Stop Solution. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 45 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending me a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point in time, I'd like to thank GenScript, who developed the content for this presentation. GenScript is the leading contract research organization in the world providing gene peptide, protein CRISPR, and antibody manufacturing services. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services, encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now, it's my pleasure to introduce you to our speaker for today's webinar, and he is Dr. Robert Ford, Field Application Scientist with GenScript Biotech in Europe. And there he is. Hi, Rob. With a strong track record in biochemistry and molecular biology, Dr. Ford brings extensive experience in various technical roles. Dr. Ford now uses this expertise to provide support with protein expression services for customers in Europe. Now, it's my pleasure to pass the mic and the controls over to Rob. So, Rob, when you're ready, you may begin.
Thank you very much for the kind introduction there, Sonia. I'm very much looking forward to talking to you all today about the about this webinar titled Enzyme Optimization and Production E. coli One-Stop Solution. I hope everyone can see my screen okay. So I'm just about to start. So what is an enzyme? So enzymes are biological catalysts that speed up the rate of a given chemical reaction. They are fundamental to all of life and have very important industrial and biotechnological applications. Most enzymes are globular proteins, but there are obviously some examples which are not. So one of those is an RNA-based enzyme. These ones are called ribozymes. And like all catalysts, enzymes reduce the activation energy to increase the rate of a given chemical reaction. The area of the enzyme where the substrate binds, so the substrate is the target of an enzyme, is called the active site. And when the active site or the enzyme, the structure of this enzyme changes, this might disrupt the active site and result in denaturation of the enzyme. So if I take an example of hexokinase, just bear with me while I get my laser pointer up. This schematic on the left here represents the hexokinase molecule, and on the right is the three-dimensional structure. Hexokinase catalyzes the phosphorylation of glucose, which is the rate-limiting first step of glycolysis. So in this process of the substrate binding in the active site, this induces a large induced fit mechanism which closes over the substrate, which allows the magnesium cofactor, shown in yellow, to catalyze the phosphorylation of the enzyme. So what factors affect enzyme activity? And how might these factors affect the kinetics of the enzyme? So in enzyme kinetics, the reaction rate is measured and the effects of the varying of the conditions can be investigated. So studying the kinetics in this way can reveal the catalytic mechanism and its role and how the activity may be controlled. So this is really important in terms of inhibitors, such as a drug that might be investigated for enzyme kinetics. So if you look at one of these factors, which is the temperature is directly proportionate to the rate of reaction. So if you increase the rate of reaction, you might get to the optimal temperature, at what point the enzyme might become denatured, and you might see a decrease in the activity. The second step is pH, and pH is important in terms of the structure of the enzyme. So for for extremes of pH, such as shown here and here, then you might see the denaturation of the enzyme due to the disruption of the chemical bonds between the substrate and the enzyme, and disruption to the three-dimensional shape of the enzyme, which is important for the catalysis. Finally, the final factor which might affect the activity is substrate concentration. So as we all know, this is a famous plot from Michaelis-Menten. So this kind of indicates as the substrate concentration goes up, you reach a level which is known as the Kmax, which is the maximum amount of substrate. So that's when there is not enough enzyme present to turn over the level of substrate, and that's this kind of method is a good way to kind of understand the function and the mechanism of action of specific enzymes. So as I already alluded to, there are many different applications of enzymes in biotechnology. So first of all, translocation enzymes are vital for the secretion of proteins in biotechnology, such as biologics. So biologics, namely mainly antibodies or with different modalities, they are kind of secreted normally by the mammalian protein expression system. Also, enzymes are very vitally important in terms of actually conjugating drugs. So for ADCs, there might be specific enzymes that are required for this purpose. Also, enzymes are very vitally important drug targets, such as kinases as an example. So kinases or these sorts of enzymes might become dysregulated in diseases such as cancer. So they are very important to be able to study and also look at in close detail. So enzymes were responsible also for the molecular biology revolution. So restriction endonucleases, DNA polymerases were kind of like the godfather of this sort of pioneering revolution. Also protein engineering, where proteins might be proteases might be used in to cleave off specific tags or other kind of modifications, such as phosphorylation. And this, you know, there are a lot of examples of proteases, such as TEV and enterokinase, which are fundamental in this approach. Enzymes are also vital for the food and drink industry. So there are enzymes such as amylase, pectinase, and lactase, which are used to manufacture food and drink in this way. For biochemical reactions, monitoring enzymes such as phosphatase peroxidase, alkaline phosphatase, and luciferase, these are used as reporters to kind of monitor biochemical reactions and are used routinely as part of IVD assays such as ELISA as kind of detection for a given analyte. Next is biofuel. There are a number of different enzymes that are really important in this industry, as well as materials. And then finally, vaccines. Enzymes play vital roles in different stages of vaccine manufacture, including being used as formulation. There are enzymes that are adjuvants and also enzymes that are used for delivery, vaccine delivery, and also the final QC of the vaccine. So really important industry for this. So what are the challenges with enzyme optimization? So there might be a need to optimize enzyme activity of a specific enzyme. There might be an example where there could be issues in terms of like need to enrich from many different candidates, and it might not have the in-house capabilities to do the high throughput protein production. So it might be more time consuming and therefore more expensive to do in-house. Enzyme solubility might be poor. How can we overcome this problem? And yes, the budget is limited, and this is far too expensive to do. And there might be a vendor to are there any help for GMP? Going from the R&D side of things all the way through to GMP. So there might be many more problems. So what solutions are there? So let me introduce you to our one-stop shop. At GenScript, we have 3 different areas where we can help you. So the enzyme engineering, we have our high throughput small scale screening and medium scale screening, and we are about to launch our new AI-driven enzyme discovery platform that can help to go from thousands of candidates to narrow down the selection of these down to tens. And then we can go all the way through to the GMP manufacture to terms of GMP-grade material. So in terms of the timelines, for the initial sort of engineering, it can take between 6 to 8 weeks. And in terms of the production, depending on whether on the type of project and what needs to be included in that process, then it can be between 6 to 9 months. So I'd like to introduce you now to our AI-driven enzyme design tool, which is going to be launched very soon, and also our high throughput platform. I'll give you a few more details about what we can do and how we can help you. So in terms of our AI-driven enzyme engineering platform, we have recently got this kind of to the stage where we are able to begin the launch of this very soon. So the customer might input a protein sequence, and from that tool we can then elucidate the structure and the properties of this particular enzyme. And then in terms of engineering, we're looking at 4 different parameters. These are stability, activity, solubility, and expression. So these might be done all in simultaneously to put into the AI platform, and or we can pick one of these particular factors and look to engineer, but mainly based on that. So from the AI, once we kind of like have the sequence, we can then engineer many different clones, and from that, on the platform, you can then basically just use that system to create the codes and initiate the production of high throughput production. So this is the kind of sequence of steps that we use to go through that process. So there are 4 different sort of steps in terms of the tool. So this is how the tool might look online once it's launched. So we'll have a welcome page, which is the AI design. So that's it, play on words there for design. And looking at different protein functions, and this is kind of the get started page. So the limit in terms of the amino acid length is between 50 and 1000 amino acids, And as I've already mentioned, there are 4 different workflows which can be run to optimize each campaign. So these properties might be run simultaneously, or you can pick the most relevant ones to the actual application of the specific enzyme, and then run that campaign in the AI platform. And you can limit the number of mutations, and then the running time is related to the length of the sequence, so it can be done in as little as 1 hour, so very rapid. And then once that is complete, then you can initiate the order, and we will then start with the gene synthesis or mutagenesis of the original clone, and provide that in a high throughput manner. So the benefits of this tool is that instead of doing the random mutagenesis and effectively screening these clones by hand, then we're able to kind of enhance the predictability and the efficiency of this process by tailored design the AI, looking at these 4 different parameters, which in turn should reduce the R&D time and obviously the cost of that. And there are many different advantages. So you don't need to be particularly proficient in IT, and this is a really streamlined process in order to generate the clones of interest to then start your enzyme optimization journey. So next on to, once you've got your specific clones, then we can go through to the small scale high throughput expression service. So the start of this is the gene synthesis and cloning step, where we can code and optimize for the E. coli expression, and also using our proprietary code and optimization tool, we're able to kind of generate high yielding clones. We will then perform the colony selection using our high throughput transformation. With this, we can also look at different E. coli strains. So if, for example, that the enzyme might be toxic, we can use a BL21D3 P-lyse strain that will reduce the kind of leaky expression that you might see with your typical BL21D3 star. From that, we can then go through to the high throughput protein expression, where we have we can use 24 well plates for the high throughput culture. So we use robotics to pick colonies. We can then do the actual sort of bacterial grow in incubators. We can either use manual IPT gene induction, or we can use auto induction media to kind of initiate the protein expression. Once we have the proteins expressed, then we go to our protein purification module, where we use robotics to purify this, normally by nickel resin, but there are other options. So following nickel purification, we would then buffer exchange and perform QC, and then either deliver the purified proteins or provide a report. Sorry, my computer's just frozen a little bit. There we go. The next, so this is just a schematic of the liquid handling workstation. So we have the Hamilton robots and also colony pickers there required for this work. So the service highlights, again, sorry, my computer's just frozen just ever so slightly. There we go. So the fast delivery in 4 weeks, including gene synthesis. We have 2 different culture volumes, so we can go from 5 mil, which excuse me, we can look at the protein expressions and provide a report, or we can use a 50 mil culture volume, in which way we can then generate enough material to purify that, provide the purification report, and then also send you the material as well. So a nice little case study here, taking T7 RNA polymerase. We took 76 candidates. We expressed this at 50 ml volume, and we had a target amount of between 50 and 100 micrograms. And we took the A280 SDS page to look at the QC. And then following the elution and 500 microliters, we got an average purity of over 90% and an average yield of 3 per mil. So overall, we were able to eliminate 10% of these candidates based on their poorer protein expression and purity levels. So we can then take those candidates forward and do more biochemical assays on them to investigate their function, etc., to then limit them even further. So the next step would be the pilot scale study. So our pilot scale production and screening, we have different size expressions that we can do. So we go for 1 liter typically, but we also have 5 liter fermenters, which are shown down here, and we can then take that to the active pilot to do the purification. So in contrast to the high throughput platform, we have additional purification that we can use following the initial first step. So because we can then do we have a larger amount of the proteins, we can also perform extra purification. So for nuclease, endotox, host cell DNA removal, etc., to then get these candidates, squeaky clean. Following this, we then have protein and report. We can then provide a CMA reports, look at the concentration, the purity, look at additional QC methods such as these mentioned here, and then finally perform functional tests and look at stability, which is obviously a very important consideration as well with enzyme manufacture. So in terms of the highlights, we can actually do hundreds of enzyme candidates today, and the reason, in terms of wanting to reduce some of the impurities, is that some impurities can affect the performance of enzyme candidates. So this is a really nice way to really hone in on the top performing candidates and really narrow down some of the targets that you want. And obviously, yield is a very important factor for this sort of screening. So in terms of the QC, these are the sorts of things that we can offer in molecular biology. We can perform these nuclease removal, endotox removals, and we can perform activity assays. So we can look at cleavage, for example, or other types of assay for polymerases. And then also we can check the host cell DNA, sort of whether we have removed sufficient amounts of DNA, which obviously might interfere with these sorts of enzymes. In terms of enzymes that we use, their biochemical reactions, we can then, obviously we can do various things as well, such as activity assays and stability assays, where you can monitor the activity over time and look at different the effects of temperature elevation on the stability over time as well. So in terms of in this case study for this pilot, we take 15 of these T7 RNA polymerase variants. We perform the expression and then 2-step purification, followed by the QC by SDS page, and then a QC for nuclease. And as you can see, in terms of the yields, we can see that there are some candidates which we can kind of eliminate, and some kind of candidates which are more promising, which might be used to take forward for further study. So in terms of the next step, which is our GMP-grade enzyme manufacturing, this is kind of the general workflow for this. So we have the initial sort of gene synthesis and vector construction, colony screening, looking at different strains of E. coli, followed by the master cell bank and working cell bank construction and stability tests, and then also upstream, downstream process development and analytical methods development. Once this is done, we then go to a technology transfer phase for the upscaling of this, and then finally the GMP grade production and QA and document preparation. So this, depending on the projects, can take anything between 6 to 9 months. So the next couple of slides, I'm just going to explain a little bit more about what we do in terms of the process development and analytical methods development. So in terms of process development with our cell banking, what we do is we can code and optimize. We then do the host screening, and then we perform look at the master cell bank, work cell bank, and the stability of these banks. In terms of process development, we will look at both the fermentation process, kind of maximize the yield. So obviously aiming for the best possible yield for each particular enzyme, and then the purification process, where we have a minimum of over 95% purity for each of this. Analytical development, where we can look at the activity and some of the other kind of components here. Formulation development, which might include which storage buffer to use, how best to perform the lyophilization, which method to keep for at what temperature, whether it's going to be frozen at -80 with glycerol, or whether it's going to be stored at 4 degrees, and then finally the transportation method, so whether it could be shipped on dry ice or whether it's not needed to be kind of shipped on dry ice. And then finally looking at different stability, so we can do an accelerated stability using the Arrhenius equation method, or look at long-term stability at kind of different temperatures to see how well they kind of hold up. In terms of the commercial production, we would then look at the working cell banks, look at hundreds of batches, make sure that each of these batches are comparable or identical. Also, we can accept work cell banks from our customers and look at how we do these multiple batches. So in terms of production, we have very flexible sort of amounts that we can produce. We have the 5 meter squared lyophilizer, and then also we will follow the SOPs and all the batch records and validate all the consistency with that. Also, we've recently obtained ISO 13485 certification, so any IVD manufacturers we can perform at GenScript to suit those requirements. In terms of QC release, then we will look at all of the kind of analytical methods that described previously to make sure that they conform to those measurements. So as a little case study here, looking at 3 different enzymes, creatinase and sarcosine oxidase. We look at these particular enzymes. So we performed the gene synthesis, the optimizer sequence, and look at the colony screening and the sort of host screening. We can then look at the different fermentation and purification, and then finally the lyophilization of these, and check that the activity is high and that there are no microorganisms present. So following this, we've done various in vitro diagnostics, synthetic biology, and GCT applications, and delivered various enzymes such as exonuclease 3, this tel-N proteomerase, and multiple different Cas proteins, MAD70 for RNA ligase, as well as these other enzymes here for these applications. And then in terms of GC to customers, then yeah, we also now have the GMP grade production service available as well. So what if my enzyme forms inclusion bodies? What can we do about that? So first of all, I just want to give a quick introduction about what inclusion bodies are. So inclusion bodies are these kind of little black dots that are seen in this E. coli. They're typically composed of insoluble and biologically inactive aggregates. So in certain cases, actually, this is kind of a protective mechanism that the E. coli does to sequester these kind of toxic proteins to prevent the harmful effects on the cellular processes of the E. coli. And they're kind of formed as a result of, poor protein folding or improper protein-protein interactions that kind of generate these big clumps and can be seen as these little black dots under the microscope. So it's really important to have a soluble protein to get highly active material. So how can we do this if the protein is not soluble initially? So the first method is that we can use high-throughput solubility tag screening. So we have multiple different vectors which have different tags that we can then drop in the enzyme into this high expression vector. And this method is usually preferred over the next method I'm going to talk about, just because you're dealing with soluble proteins. So so if you imagine this backbone vector with each tag in, we can drop this our protein in with different tags, and then we can do a high throughput screen. So say we had 10 enzymes that we're looking at which had poor solubility. We could then take those and add them to these tags and look at the expression versus the wild type. So as you can see here, the wild type has very poor expression. So these 4 tags therefore show the expression in the whole cell lysate and then also in the supernatant. So for example, maltose binding protein would be a very good candidate for taking forward with this example. Our second solution is called our professional refolding service. So the way that we do this is we would normally induce the bacteria normally. Then when the inclusion bodies form, we can isolate these. We can then wash the inclusion bodies and then resuspend in water, and then using our variety of different refolding buffers, then we can check which ones work best in terms of the purification. So we can look at then upscaling the preferred refolding buffer and check for activity. So a nice little case study was performed using this protein called RHBMP9. So this is a very challenging protein. It has 3 inner chain disulfide bonds as well as an interchain disulfide bond. You can get expression in CHO, but it's very, very poorly expressed, so not really feasible to scale up. So what we were able to do was initially we were not getting any expression, and then by using our professional refolding platform, we were then able to get 73 milligram per litre worth of material, so that with very, very high purity. So that represented a very nice result. So in terms of, if you have a problem and you need to increase the activity and then scale up for GMP production, hopefully I'll convince you that we've got your back with this. So, we have the newly launched AI platform, which can then feed into our high throughput platform, which has a very large capacity. So we're able to purify lots of different proteins simultaneously. We have, very fast turnaround time for this. So for the initial sort of high throughput, we can do this in 4 weeks, and then for GMP production, we can get this done in around 6 months. And we have a variety of different ways that we can be flexible. So at any stage during this process, we can kind of help at any different point. So if you're not at the start, you just were looking to kind of look at the GMP production, then we can kind of join at that point, or we can help with the the overall optimization of the protein expression, or we can kind of help with the refolding as well. So we can be very flexible. in our approach to help with the customers. So thank you very much for listening, and I invite Sonia to kind of help with taking back control. So yeah, thank you very much, everyone.
Okay, well, thank you very much, Rob, for that very insightful and engaging presentation. I hope everyone enjoyed it, and I'll invite Rob to put on his webcam. There he is. All right, perfect. So now we are jumping into the Q&A portion of the webinar. I did receive a bunch of questions while you were speaking, Rob, so I'll start with those. But just a reminder to our audience members that you still have the opportunity to send in your questions now. So please go ahead and do so by using that chat box, and we'll try to attend to your questions during the time that we have together. Now, let's start with this first question here, Rob. This audience member is asking, the AI enzyme optimization platform sounds very interesting. When does it launch commercially? Good question.
Oh, sorry, yeah, I meant to actually mention that in the presentation. Thank you very much for bringing that to my attention. Yeah, so it's meant to launch at the end of this month. So it should be on your radars by the beginning of October. So hopefully very soon indeed.
Okay, perfect. Thank you Thank you very much for that question, and thank you for that answer. Here's our next question. A nickel NTA purification is great, but what if what if I don't want to have a his tag on my enzyme? What other options are available for purification? Is it possible to remove the tags following purification, for example?
Yes, thank you very much. There are a lot of options. So in terms of our high throughput platform, you don't have to use the nickel NTA method of purification. We also have GST resin as well as flag resin. So we can use those sorts of tags to do the initial purification. In terms of like the pilots production, we have many other varieties of steps that we can do. So we have ion exchange, for example, hydrophobicity, interaction chromatography. We also have these other tags that I've already mentioned, and also size exclusion chromatography. So we have various different methods, and it will just be tailored to the unique requirements of the customer. In terms of the tag removal, yes, we can do tag removal as well. So, you know, if you wanted to remove the tag, we could have the tag at the end terminus and put in a protease cleavage site. And then we also can do the protease cleavage in-house for you, or, you know, obviously ship you the material for you to be able to do the cleavage as you request. So there are those sorts of things that we can do. We also have sumo tag and we can sumo cleave as well. So there are many different options, and each option is very customizable. So we will look to do this dependent on the customer's needs.
All right, thank you for that. I am paying attention, but I'm also paying attention to all the questions that are coming into the chat box here. So everyone bear with us. We'll get to them shortly. Let's go to the next question here. This audience member is asking, I'm interested in the HTP enzyme expression service, but I already have the plasmids. Is it possible to ship the plasmas of candidates I'm interested in? That's a good question.
Yes. Yeah, absolutely. So if you know, you already have the plasmids available, we don't need to do the initial gene synthesis. You can indeed send us your plasmids via you know, we have different offices located in the Netherlands, the US, or the UK, for example, where if you send the plasmids to the office, we have the internal logistics team that can source the rest. So it's very seamless transportation from you know, for your plasmids for us to work directly with them. So all we request is a few micrograms of plasmid for each candidate, and what we will do is we'll do the QC on those plasmids, make sure the sequences are as expected, and then perform the high throughput protein expression service. So yes, of course, that's possible.
Okay, thank you so much for that. We'll go to the next question here. For AI-driven enzyme discovery, does GenScript claim IP ownership of the enzyme, or does the client have IP ownership?
Yes, the client has the IP ownership of that.
Okay, perfect. Thank you. In the case of studies, what is the general cost for each segment? Design, fermentation, purification, formulation?
Um, I'm sorry, can you just repeat that one? I
Yeah,
just didn't quite catch it
in,
yet.
no problem. In the case studies, what is the general cost for each segment?
Oh,
Design,
costs.
fermentation, yeah, purification, and formulation. Sorry about that.
That's okay. So typically what we will do is it will be done on a case-by-case basis. Obviously, each case is highly tailored to the customer's needs. So what we would suggest is if the customer would like to get a quote, then get in touch with one of our representatives, and we can organise a call. We can go through each of the options, and then we can look to do this costing. It's as a field application scientist, costing is not part of my remit, so I apologise for that.
Okay, we get to the next question here. Um, okay, this one here, 3 questions actually. This person is asking, how many variants are typically suggested by the AI tool is 1. So we'll start with that, and then I'll go on to the next ones. Or do you want me to give
Yeah,
you all 3 of them?
no, that's fine.
Okay.
So typically, I think in the region of 10,000 can be applied, but obviously they will have a ranking-based system, so you can
Mhm.
kind of choose the ones which are the most sort of interesting from what the AI tool has sort of enriched. So it's very much a custom user, custom friendly sort of way to kind of however many sort of candidates you would like to look at, um, and yeah, it will give you a ranking based on what it thinks from those parameters that you're interested in. So again, it's kind of very much dependent on the sort of capability and the number of candidates that the customer is interested in.
Okay, all right. We'll go to the next part of that question. They're also asking who holds the IP of a variant that was developed by the AI tool.
Yeah, again, so the IP is the customer's IP if they want to take that forward.
Okay, and then the final question here that this person has is how would you deal with enzymes that are not expressed in E. coli?
So in terms of other enzymes, we have other platforms.
Mhm.
So we have the insect platform. So if you have a more challenging enzyme such as like a kinase, we would typically look at using our insect platform for that. So we do have an insect high throughput platform, which we can also use for the production of these sorts of enzymes. We also have a mammalian platform, so our TurboCho platform is newly launched, and we can look at non-mammalian non-E. coli based proteins using this platform as well potentially. So there are different options for post-translational modifications and more difficult to express proteins, etc.
Okay, and we'll jump to the next question here. Please, what kind of protein is inside it? That's
Sorry,
all
can
they
you
wrote.
repeat that?
This person just wrote here, what kind of protein is inside it? Because enzymes are proteins, but are all proteins are not enzymes?
Yes, of course. Yes, absolutely. So, we can look at a variety of different proteins using our 3 different platforms. This talk was specifically about enzymes, but we have various different sorts of proteins that we can express and purify using our services. So, for example, using our TurboCho platform, we can use this to express various different antibodies with different modalities. So we can look at fragments, we can look at bispecific antibodies, and all sorts of different based sorts of therapeutics. Looking at our high throughput platform, we then can upscale that accordingly using our different services such as our TurboCho Express and our high performance platform. We can look at other sorts of proteins such as T cell receptors and other sorts of ligands as well. So yeah, we can we have a large level of expertise in order to kind of do many different sorts of proteins.
Okay, fantastic. Thank you so much for that. Okay, do you also have a HT platform for other protein expression systems?
Yes, as I already alluded to in one of the answers to my other questions, we also have the insects platform as well as our mammalian platform. So we have our turbocho platform, which is newly launched as well. So yeah, if you are needing any proteins which have needing for post-translational modifications, etc., then we can use these other platforms for that.
Okay, all right. I'll jump to the next question here. I'm interested in GMP grade production of a protein that is expressed in E. coli, but it isn't an enzyme. Is this something that you can also accommodate?
Yeah, thank you for bringing that up. Yes, we can also look at GMP grade material which isn't enzyme based. So, for example, we have experience with production of insulin with our GMP production. So obviously this needs to be refolded. So we have a large capability and big amounts of these refolding reservoirs that we can use for this process. So we can also do these types of proteins as well as others. So again, it will be dependent on a case-by-case basis, but we are able to accommodate different sorts of proteins as well. Yes.
Okay, all right. Is the system optimized only for E. coli, or can it be used for other bacterial hosts as well?
So we have got many different strains of E. coli. Mainly, it is optimized for our E. coli platform, but we have also used Bacillus before. But yeah, we would need to kind of check in back with the production team whether we can use the high throughput service with this. So yeah, if that customer wants to get in touch with us directly, then we can get a bit more of a solid answer for that question.
Okay. Is it possible to optimize more? I'm sorry, let's say that again. Is it possible to optimize more than enzyme pathway using your service?
So is this? I'm assuming this means the AI tool. So this is for enzyme optimization. It's not for other sort of protein optimization, is my understanding.
Okay. All right, let me go to the next question here. Instead of gene synthesis, I'm interested in creating a library for screening many enzyme candidates. Is this something that GenScript can also do?
Yes, absolutely. So as you're aware, we have our gene synthesis platform, but this platform can also be utilized to make precision mutant libraries, for example, and we have high throughput mutagenesis service as well. So there are different ways to kind of skin a cat, if you like. So we can create these clones using these different methods, but it will depend on sort of like what the most time-effective way is and what the most cost-effective way is, depending on the sequence that are kind of given from the output. So there are numerous ways that we can do it, and we have that sort of capability to kind of look at the sort of best way that is, we can do it and do that accordingly.
Okay, all right. All right, let me just jump to the next question here. All right, I'm concerned about imidazole carrying over effective enzyme activity during enzyme HTP screening. What can you do to circumvent this? Imidazole.
Imidazole, is that?
Yeah.
Yeah. So yes, so imidazole is used to elute the histag proteins from the nickel NTA. So what we can do is we can perform multiple buffer exchange steps to actually make sure that there is a negligible amount of carryover. The other thing that we can do is obviously use different tags for this high throughput method. So we can use GST or flag tags as part of the purification process. So there's no need at all for imidazole if that is kind of a primary concern. So there are definitely options that we can do to kind of circumvent that.
Okay. And is there an option to build in a functional assay following HTP enzyme production?
Yes, absolutely. So we have the capability to kind of transfer assays from the customer into our production team. So if there is a functional assay that you would like us to do following our high throughput production to kind of eliminate sort of some of the clones at this stage without having to then ship them to you, then that's definitely something that we can incorporate as part of our workflow.
Okay, all right. It looks like we have two more questions here as we're coming to the ending of the hour. This audience member is asking, in the case that the enzyme needs cofactors, fat binding, does the AI consider particular cases? Also, the system can optimize for protein stability, for example, at room temperature.
That's a very good question. I'm not actually sure of the answer to the first part of that question in terms of the cofactors. So again, I will be interested to kind of find that out myself. So yeah, please do get in contact with me, and we can talk to our technical team about that in more detail. And then in terms of the other part of the question, in terms of the temperature, yeah, so stability is one of those factors. But again, whether that's kind of with the cofactors taken into account, then that's something I'm not 100% certain about. So yeah, good question.
Okay, all right. Let's see if I can grab another question here. Does GenScript have experience with cytoplasmic expression of enzymes?
Absolutely, yes. So that's the main method that we would express enzymes is cytocyclically. So we would not look to make the enzyme soluble in order to do the purification, especially within the high throughput platform. You can't really look at insoluble enzymes using our high throughput platform. So they would be a case of from the 1 liter pilot study stage, doing the refolding method. So we would always look to make the protein soluble at that high throughput stage if possible. So the first kind of step would be that tag screening step, as I kind of mentioned earlier, to kind of look at that sort of insoluble. But yeah, normally we would take the soluble protein for that purification.
Okay, all right. Okay, this audience member is asking, is there an option to build in a functional assay following HTP enzyme production?
Yes, absolutely. I think we've already had a very similar question to that, and the answer is yes. We can build in a functional assay to kind of help with the sort of narrowing down of candidates following high throughput protein expression.
Okay, we're really getting all these questions too. I've never seen so many questions come in. This is great.
Great.
Looks like we're narrowing down. This might be our last question here. This audience member is asking, do you have expression system that do not require antibiotics?
Um, I don't think so. I think all the yeah. So again, I'm not 100% sure whether that's
Mhm.
something that I know. I think maybe the mammalian systems don't require antibiotics, but again, I'll double check that and get back to you.
Okay, well, thank you so much, Rob, for answering all those questions. And thank you very much for that very engaging presentation. I think everybody enjoyed it. I just want to say thank you very much to our audience for sending in all those questions. We truly appreciate it. We have reached the end of the Q&A portion of the webinar. For those of you who are interested in hearing more about GenScript, you can register for their next webinar. And there it is. So please go ahead and mark your calendars for the next webinar that's slated for Friday, October the 6th. And I'm actually going to put the registration link in the chat box right now so that you'll have that handy to make it very convenient and easy for you to access. So it's right there, everyone, for you to check out. All right. And if we couldn't address all your questions, because there was a lot of them that have come in, and Rob will be more than happy to answer them. So please go ahead and utilize his email address here on your screen if you have any further questions or need more clarification. He'd be happy to assist you. Okay, well, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X Talks with access to the recorded archive for this event. Additionally, there is a link to view the recording of this event, which you can also share with your colleagues so they may register for the recording here as well. Now, a survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars and get even more out of today's presentation by downloading the supporting materials that we have available for you under the handouts tab. And you'll see that we have two brochures there for you. So please go ahead and check that out. I encourage you to do that. Now, please join us in thanking our speaker, Rob, for that very insightful presentation. So thank you, Rob.
Thank you, Sonia, and thank you everyone for listening.
Okay, great. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, Rob. Bye, everyone.